So this is what I've been building up to with my neuroscience and genetics series, a technique called single cell sequencing. This is a relatively new technique that's taken off and probably in the next 10 years is going to garner a Nobel Prize for some part of this. It's totally changed the way we've been able to look at cell types. And so here's a little bit. I've talked a little bit about what makes one cell different from another. It's that it expresses different genes, right? And so in older techniques for sequencing, basically it would look like this. You'd have some tissue sample of a bunch of different types of cells. They express some RNA. We would grind them up, have the RNA, and we would sequence it. And then we would get to see, okay, these are the levels of the, in this case, red, blue, and green genes that we have in this tissue. So the best thing or assumption that we can make is, oh, all of these cells on average express the same level of red, blue, and green, when in fact that's not the case, right? So what we do with single cell sequencing is there's a number of different ways to do it, but all of them have this step where each cell or nucleus is isolated in a little droplet, so that when these cells in this tissue sample are isolated into their single droplets, we actually put a little genetic barcode into each individual cell that when it comes out on the other side, we can use our cool little genetic tools to read all of the RNA and see which RNA is from the same cell and which is not. So when it comes out on the other side, we can say, oh, all of the cells that were expressing the blue gene did not express green or red. And the same thing for all the others. So that when we analyze this data, we can say, oh, we actually have 3 different types of cells here expressing just the blue genes, the green genes, or the red genes. And that's a totally different picture than you get if you are just saying, oh, all of these cells are the same and on average. So it's a really powerful tool, and in my field in neuroscience, it's changed the way we think about neurons and the way we think about different types of neurons and what they're doing. And it's allowed us to look at how all of the different neurons are changing with disease processes or interventions. So it's a very cool technique, and I do hope you look into it because it's really neat, really fun, and I hope you learned something.